Now, all right, next up we're going to talk about active transport across the plasma membrane. Active transport, there are going to be times when a cell might need to increase the concentration gradient across the plasma membrane in order to accomplish more work. The process of moving molecules against their concentration gradient is called active transport. This is where we're moving from an area of low concentration to high concentration. This is always going to require the aid of carrier proteins to move those molecules across the membrane. Active transport also requires the input of energy, typically in the form of ATP, to help carry that across the membrane. Great example of this would be the sodium-potassium pump. Proteins involved in active transport are often called pumps because of their similarity to water pumps that require energy to push water against the flow and force of gravity. One type of pump that we're going to talk about because it's active in all animal cells but is especially associated with nerve and muscle cells would be the sodium ion pump. It moves sodium ions to the outside of the cell and potassium ions to the inside of the cell. The transport of sodium and potassium is linked together through the same carrier protein called the sodium-potassium pump, and it carries these molecules by undergoing conformational changes in order to bind those two ions. So here's an example of what that looks like. The sodium-potassium carrier has an initial shape that allows it to bind to 3 sodium ions. These are going to move in to these spots here. Now, once they move in, a phosphate group is going to be broken off ATP. We're going to turn ATP into ADP. We're going to break that final phosphate bond and release a burst of energy that is going to allow this potassium pump to change shape. And when it changes shape, it's going to open up on the other side, and those sodium ions are going to move out of the pump. So we're moving from an area of from low concentration to high concentration. Now, once we are opened on this side, potassium ions are going to then bounce into these carriers. Now, this particular pump can carry 3 sodium for every 2 potassium. Once those potassiums settle into their locations on this carrier protein, we're going to undersee another conformational change. It's going to close up on one side. We're going to release that phosphate group, and we're going to open back up. Now, once we've opened up, 3 sodium ions can move in. We're going to attach an ATP. We're going to break off that phosphate group, release a burst of energy, allow that conformational change, release the sodium ions, and allow potassium to fill back in the gaps. Once potassium has filled in, this carrier molecule will snap shut, release the phosphate head, and then open back up again to release the potassium. Sodium can now bind in, ATP will break, release sodium on the other side, so on and so forth. The cycle continues. So that is our sodium-potassium pump. Another really great opportunity for an extra credit video if you want to send me a video on how the sodium-potassium ion pump works. So this is how we're going to move small molecules. Small molecules can move across the plasma membrane with the aid of carrier proteins. Very, very large macromolecules, like full-on sized proteins, are going to be a lot harder to move. These are going to be typically transported into or out of the cells in bulk transport. We're going to use 2 methods, exocytosis, which is how we get macromolecules out of the cell, and endocytosis, which is how we get macromolecules into the cell. Exocytosis, the way we're going to do this, as we kind of have already mentioned with the transport of proteins. Proteins are going to be synthesized by the ribosomes in the endoplasmic reticulum. They're then going to be passed in transport vesicles to the Golgi apparatus. Once inside the Golgi apparatus, they're going to be packaged, tagged, and shipped to wherever they need to go, either somewhere else in the cell, or they're going to undergo exocytosis and be released to the outside. That transport vesicle is going to come to the surface of the cell. It's going to merge, it's going to fuse its membrane with the plasma membrane, and when it does so, it's going to release its proteins to the outside world. Endocytosis looks like the exact same thing, but in reverse. So in exocytosis, those vesicles are going to fuse with the plasma membrane to secrete those contents. In endocytosis, the cell is going to engulf substances into a pouch, which will then become a vesicle, which can then be digested. This is what endocytosis is going to look like. Endocytosis comes in 3 forms depending on what it is we need to do. The first type will be phagocytosis. Phagocytosis occurs when the material to be taken up is large, like a large food particle or another cell. What's going to happen is the plasma membrane is actually going to reach out and wrap around that particular macromolecule or cell. It's then going to pinch off that vesicle and bring it into the cell. Once inside the cell, it can break it down and use it for whatever purpose it needed to phagocytize that. And this is going to be very common in single-celled organisms like amoeba. It also occurs in white blood cells in humans. This is how our white blood cells can actually clean up after an infection. It can engulf debris such as worn-out cells. It can also engulf viruses. It can also engulf infected human cells. So phagocytosis is a very important part of our immune system. Pinocytosis, on the other hand, looks very, very similar. But in this case, we are not taking in large materials, we're taking in lots of small materials, typically multiple small materials or a liquid. It requires a special electron microscope to see pinocytotic vesicles. These are very, very small. Phagocytosis is very large. An extra bonus point this week for anybody who sends me a video of a white blood cell hunting down and phagocytizing a bacteria. There's videos out there, great live videos of where you can actually watch a white blood cell in real time hunt down a bacteria, engulf it, phagocytize it, and then start breaking it down to digest it. Pinocytosis is a lot harder to see. I would also appreciate it if anyone could send me a video of pinocytosis happening. It is going to require a special electron microscope to see this happening because these vesicles are so incredibly small. We're taking tiny little sips here. We're not biting off a great big giant steak. We're taking tiny little sips here. So it's also sometimes referred to as cellular drinking because we're forming tiny little vesicles around a liquid or very small particles. Now, there is one other form of endocytosis that we can use, and that would be receptor-mediated endocytosis. This is a special form of pinocytosis that requires receptor proteins to recognize materials to be brought into the cell. In this case, molecules such as vitamins, peptide hormones, lipid proteins, lipoproteins can bind to specific receptors. These are going to be found in special locations on the plasma membrane called a coated pit. And the reason we call it a coated pit is because of the layer of proteins on the cytoplasmic side of the vesicle. This coat of proteins is going to be bound to receptor proteins that are going to only bind to and recognize very specific molecules. This is a very highly selective and very efficient method for targeting specific materials that you want brought into the cell. That's it for active transport. All of these methods, and I'm sure, unfortunately, I didn't really specify. These 3 methods of endocytosis and exocytosis are all going to be controlled by the cytoskeleton. Every movement of that cytoskeleton is going to require huge amounts of ATP. That's where the energy comes into play. This is where we're moving areas from not just moving against the concentration gradient. We may not even be moving against the concentration gradient, but we're just trying to get molecules that are way too large to be just transported across the plasma membrane. They're going to have to go through endocytosis. All of this is going to require the involvement of the cytoskeleton, and just like it requires energy for you to move your arm or wave your hand or pick up that sandwich, it is going to require quite a bit of ATP in order to reach out and engulf these macromolecules. So that is it for active transport across the plasma membrane. The last section that we're going to cover will be the modifications of the cell surface.